This study aimed at testing the hypothesis that the main neuropathological hallmark of Parkinson's disease, which is the aggregation of the alpha-synuclein protein, could start in the gut and then spread to the brain through the so-called gut-brain axis. For that, we injected baboon monkeys with alpha-synuclein aggregates purified from the human brain of Parkinson's disease patients. The injections were done intragastrically in the stomach and in the duodenum, and the brains of the monkeys were analyzed 2 years after the injection for the presence of alpha-synuclein pathology and signs of neurodegeneration. At the same time, another group of monkeys were injected intracerebrally in the striatum to test the possibility that alpha-synuclein pathology could also spread from the brain to the gut. The results from these studies showed a bidirectional route of propagation for alpha-synuclein from the gut to the brain, but also from the brain to the gut, providing for the first time experimental evidence in non-human primates of the importance of the gut-brain axis in the initiation and propagation of Parkinson's disease pathology. After this study, we are continuing to investigate the different possible routes of propagation for alpha-synuclein from the brain to the gut and vice versa, and we are also testing different therapeutic strategies aimed at interfering with this propagation.